Major depressive disorder is a psychiatric disease characterized by a distinct rise in mood such as sadness, irritability, and bereavement, accompanied by several psychiatric and non-psychiatric disorders. The development of MDD is unknown, as it is a complex heterogeneous disorder with several contributing factors. The gene most associated with depression is the serotonin transporter gene. Serotonin is a neurotransmitter scientifically known as 5-hydroxytryptamine and is part of the monoamine group containing amino acids that play a role in many functions such as decision-making, emotions, happiness, and rewards. Originating mostly within the brainstem amongst a cluster of nuclei called Raphe nuclei, and once synthesized, serotonin is sent to the nucleus accumbens, then to the hippocampus, cerebellum, and spinal cord. During a normal neurotransmission, serotonin is released into the presynaptic cleft, partaking in one of three functions, one of which being transported back into the presynaptic neuron by the serotonin transporter to help with the next nerve signal. Low levels of serotonin are caused by a genetic polymorphism present within the promoter region of the serotonin transporter gene, known as serotonin transporter linked polymorphic region, short ALL. Genetic polymorphism means that at a specific location in the genome causes a slight variation in the DNA. A person carrying a combination of two or more short alleles within the promoter region of their gene are more at risk in developing depressive symptoms, because short alleles reduce the number of serotonin transporters within the presynaptic neuron, causing a reduction in serotonin within the synaptic cleft. Transcranial Direct Current Stimulation, abbreviated as tDCS, is a non-invasive brain stimulation paradigm modulating cortical excitability primarily through the resting membrane potential via externally alternating current stimulation and direct current stimulation. tDCS delivers a weak direct current to all compartments of local neural activity, targeting cortical regions and stimulating surrounding networks. tDCS has demonstrated to be a valuable and promising form of treatment used within neurophysiological, cognitive neuroscience, and clinical domains, with the pleasing addition of being cost-effective, easily portable, and well tolerated, from minor side effects of slight itching under the electrode, headache, fatigue, and nausea. The beneficial effects have shown in a wide range of diseases such as neurological conditions, psychiatric indications, and pain conditions. The stable weak direct current is delivered from a constant current stimulator and through to two service-conductive rubber electrodes known as anode and cathode, sized between 25 and 35 centimeters squared. The electrodes are specifically placed over the scalp in accordance with the 1020 electrode EEG placement system. The current intensity varies between 1 and 3 milliamperes and is commonly applied between 10 and 20 minutes. However, the intended duration and strength of application is dependent on the individual and disorder type. The cortical excitability during stimulation begins with polarity-dependent shifts in the neuron's resting membrane potential. The weak direct current enters through the anode electrode, causing an excitatory effect in depolarization, and exits through the cathode electrode, causing an inhibitory effect in hyperpolarization of the neuron soma and basal dendrite. Physiological alterations is achieved through sustained stimulation within a matter of minutes during treatment, lasting more than 7 hours to more than 1 day after stimulation. Dating back to the 1960s, tDCS has been used as a non-pharmacological treatment option to generate modifications of cortical excitability in preclinical studies and as a therapeutic tool for MDD. In recent years, tDCS have been found to have antidepressant effects and beneficial effects on cognitive function by regulating neuroplasticity, by increasing the serotonin levels in the synapse through blocking serotonin's reabsorption back into the presynaptic neuron with selective serotonin reuptake inhibitors, abbreviated as SSRI. Hypermetabolism is cited mostly on the left dorsolateral prefrontal cortex, abbreviated as DLPFC, associated with the right prefrontal hyperfunction in MDD patients. Therefore, the placement of the anode electrode is typically placed over the left DLPFC and the cathode over the contralateral supraorbital area corresponding to F3 and FP2, in accordance with the International 1020 electrode EEG placement system. One notable study demonstrated positive results on the efficacy of 5 sessions with 1 mA for 10 minutes in 10 MDD patients. The replication of this study demonstrated an average reduction of 40.4% in the Hamilton Depression Rating Scale score with 2 mA for 20 minutes in 40 mild to moderate MDD patients, in comparison to a 10.4% reduction from placebo stimulation. Another study showed no superior antidepressant efficacy in 10 sessions with 1 mA for 20 minutes. However, in another study demonstrated a greater reduction of 28.4% in the Hamilton Depression Rating Scale after 15 sessions with 2 mA for 20 minutes in 64 unipolar and bipolar depressed patients, in comparison to a 15.9% reduction from placebo stimulation. Another notable study demonstrated positive results from 10 sessions with 2 mA for 20 minutes in 40 MDD patients with an average reduction of 42.1% in the Beck Depression Inventory score, in comparison to a 15% reduction in fluoxetine. tDCS may be quite effective to treat MDD and to improve the condition of MDD patients. To date, the improvement of attention and working memory has been reported after 1, 5, and 10 sessions. Positive results have also been observed in other cognitive domains, such as cognitive control, processing speed, and in emotion recognition. tDCS can be a relatively safe method with few adverse effects. However, further research is required between the correlation between tDCS and MDD, with questions regarding optimal stimulation parameters, task impact during tDCS sessions, and possible influences from add-on medication during stimulation, as well as the inter-individual variables regarding anatomical differences, genetic factors, personality, comorbidities, cognitive strategy, lifestyle, and baseline neuronal activation state.